I'm going to talk about water quality, and we're focusing predominantly on Dublin Bay, but what I'm telling is, or what I will explain to you is probably applicable to many different environments. Um, so the project that I will tell you about is the Climatize. Climatize has two iterations. The first one was Interact project that ran for 6 years, which was funded through the EU with a number of stakeholders in Ireland. And then we had partners in Wales, Aberystwyth University, under the leadership of David Kay. Some of you might be familiar with him. That project came to an end, and then we had the sequel, Climatize 2, and hopefully the sequel is better than the original still. This is funded by the Bathing Water Task Force, which focuses specifically on Dublin Bay, and again, you can see it has a number of partners and stakeholders. So they're focusing on water quality, and the idea there is to do two things. We want to do to protect public health, and the other thing is to analyze where pollution is coming from so that we can have evidence-based and targeted interventions. If you ask anybody in Dublin where pollution would come from, people would predominantly point their finger to the Ringsend wastewater treatment plant, and that's it. Or some people blame birds or whatever it is. We need evidence-based, and that's what we have provided in these two projects. Now, the projects involve a large number of fieldwork activities, not just by ourselves, but every summer we are joined by a number of students from universities around Ireland. As you can see here, they're very enthusiastic people. We ship them off on boats early in the morning and leave them on the water for about 12 hours, well, 6 hours, and then we change. But they do all kinds of very useful activities on both at sea and also in the catchments to generate the data that we need to analyze where pollution is actually coming from and what can we do about it. Now, the projects involve things like this. So we measure, obviously, fecal pollution, because I'm predominantly talking about fecal contamination. But we also need to identify the biological source. We need to instrument rivers and streams and catchments to measure how much water is coming through this. We need to do source apportionment. So we need to know how much is coming from where. And then we need to model. We need to have connectivity. If you release pollution at one location, will it go to another location? So are they connected? And that's basically what I will show you. And here you see some of the instrumentation that we have, including a laboratory canoe, which was extremely useful. So how do we go about this? So in Dublin Bay, we divided it in 4 clusters. Every cluster had 4 points in it. And then we sampled these 4 clusters during the summer, during the bathing season, on the high tide, low tide, spring tide, neap tide, wet conditions, dry conditions, and then measured a whole bunch of parameters that are involved in, or that are indicative of water quality. In addition, we measured the movement of water, and all of this data can then be used to model what's going on in the bay. Now, if you look at the water movement around Ireland, you can see that further in the sea, the water just moves south and north, so that is the normal tidal movement. But if you look carefully, you see that in Dublin Bay, things are very different. That is because there is a lot of input from water. Dublin has about 60 streams and rivers, but then also the water moves differently in the bay itself. So things might not be as straightforward. People tend to see water bodies as homogeneous. So you take one sample, and actually the Bathing Water Directive actually assumes that. Take a single sample that will tell you the quality of water. And I hope to convince you that this is not true. So let's look at this and take this model. And so what can you do? You can model the tracking of pollution released from various sites. So what you see here is Dublin Bay. You may recognize it. This is the Ringsend treatment plant, Dunleary, and so on. And we're going to release pollution from the Ringsend treatment plant and the Liffey and see what happens. And if you do this, you see that the pollution is actually moving northwards. It goes into the bay itself and then swings north around Howth towards Portmarnock, but it does not affect the southern part of the bay at all. So there goes our first thing out of the window. The water is not homogeneous and things move differently in different locations. So let's do the same experiment again based on the models that is based on actual data. And now we're going to release pollution from a very small stream here and see what happens then. So this is in the southern part of the bay. And when you do this, you see a completely different result. In this case, the pollution doesn't really make it out of the bay at all. So the tides are moving this back, which is due to this complex water movement in the bay itself. So when you look at these models and data, you see that the northern part of the bay and the southern part of the bay are impacted by completely different things, which is a very useful insight. The other thing is that the deep end of the bay itself usually has excellent water quality, which suggests that pollution events, certainly in the southern part of the bay, is near shore, not offshore. Now, to look at that, you can ask yourself, what are the pollution sources in Dublin Bay? So this is what I just showed you. So you have the release of particles here. We see that moves along Dollymount and then around Howth in this direction. If we move pollution here, it gets stuck here in the southern part of the bay, which is Sandymount and Marion Strand, and then this part of Dunleary. So we need to find out what the pollution sources are and how much of each, is each source contributing. So let's have a look. Now, the way we do this to identify the biological sources, there are various ways of doing this. One thing is just to look. If you see a cow in a river, you don't need any molecular techniques to tell you the cow might be polluting. However, very often you can't see exactly what is in the water. Now, for this, we make use of the microbiome. So the microbiome is the intestinal tract of animals and humans. We all have a large amount of bacteria in your body. Actually, you have more bacteria than human cells, believe it or not, 10 times have more bacteria than human cells. And when we look at the microbiome in these animals, we see that they are different. That, of course, depends on the lifestyle, diet, and so on. So there are some bacteria and other markers that are unique to, let's say, a horse or a pig or a cow or a dog or a human. And we can isolate these and quantify these with a technique called qPCR. You might be familiar with this. So this is microbial source tracking. There are other ways of doing it, tracking, for example, specific chemicals, because it's well known that, let's say, cows don't really use coffee. So that's another way of doing things. Now let's look at the southern part of the bay. So this is more Dollymount and Sandymount, Marion, the Dunleary area. And what we did there is do a lot of sampling every half hour through a tidal cycle, and we ask ourselves what happens. Now this is the summation of all of the data, and you see it varies quite a lot. And if you look at in the day itself, day 1, day 2, day 3, these are single days, and you look at the spread of these things, you see that there is a huge variation in the levels of fecal indicator bacteria in the day itself. And it goes from very low, this is excellent water quality, to very high, poor water quality. So within the day, you can go swimming at one location, and you can come in the morning and you swim in excellent water quality. In the afternoon, the water quality will be poor. And this is something we've seen everywhere, not just in Dublin Bay. Our colleagues in Wales have seen this as well. Huge variations during a tidal cycle. And that means that the current protocols for classifying water quality could do with an update to take this into account. And of course, it has implications for public health as well, that you want to predict when poor water quality is happening. So let's look at this and ask ourselves, where does it come from? So this is an approach that you can basically apply to any type of catchment or any type of bathing water that you're interested in, and we've done many of them. This is Dublin Bay. So here you see the southern part of Dublin Bay. To compliance point, this is where the local authorities, in this case, Dublin City Council, will take samples for the bathing water directive to classify water. And if you look at the levels there, so this is Dollymount, Sandymount, and Marion Strand. You can see the pollution levels vary from very low to quite high. And here you see EP is the Elm Park stream, which is this stream over here, and this is the Trimbleston, which is this stream over here. And you see that the levels of fecal indicator bacteria are very high. And in the Elm Park stream, up to 500,000 per 100 mils. This is pretty much raw sewage coming through. And this is an extremely small stream. You would never guess it had such an impact. And with our techniques of source tracking, you can ask yourself, what is the source of this? Where does it come from? And this is a human marker. I won't show you the others, but the human marker is exceedingly high in these two streams. And for comparison, this is the outfall of the wastewater treatment plant at Ringsend. These levels are much, much higher. So this pollution here is human. So that is a major source. So does that explain? Well, no. Well, I have to do this first, sorry. You can ask yourself, where does it come from? This is the catchment area, and you can look and see that there are various locations where we sampled. And when you look and take an underground map of these sampling locations, and you go back from the sea towards the upstream areas, you can see that the water pollution is quite high in the stream until between here, and then it goes from very poor to excellent. And here is a combined sewage overflow. And indeed, along this entire stream are pipes that discharge. So you can locate where the pollution is actually coming from. Now, this type of approach you can do, of course, in other catchments, and indeed we have. Now, that is human. Now, what about the birds? Okay, you can do this too. This is, again, a transect, but this is a very rapid one. So we go out and sample this within an hour, so at high tide, and you go away from the beach deeper, about 2 kilometers, and you see that the water quality is changing dramatically. Close to the beach, the water quality is sufficient, and then you go to poor water quality, about 1,000 to 2,000 CFUs per 100 mils, and then as you go deeper again, further away, the water quality becomes excellent. So it is not homogeneous neither in space, because it changes within the day, nor is it homogeneous, sorry, in time, nor is it homogeneous in space. You go you can go from excellent to poor within a few hundred meters. Now, where you see this spike, you also see the spike of the bird pollution. So this can be attributed to birds foraging here, and anybody familiar with this area knows that there are quite a lot of birds. Then what about the other pollution there? Sorry, you can estimate how much there is. So this is data from Birdwatch Island. So you can look at the total number of birds in this area. We know the average weight of a bird poo, believe it or not. We also know how many CFUs there are for E. coli and intestinal enterococci per gram of bird feces. We know how often a bird actually defecates, so very interesting. And from that you can actually calculate the loading, and that of course is serious. So birds do have an impact, especially in September, where there are a lot of migratory birds, but not too much. Then what about dogs? Now, Sandymount is very popular with dogs. We did some sweeps. This is 9 different days. You have people on the beach, just running through the beach and measuring dog poos with the GPS in their hand. Again, we know the average weight of a dog poo. We also know how many CFUs there are per dog poo, and we can calculate loading. This is quite worrying. These red dots are dog poos on Sandymount and Marion Strand, with an average of around 1 to 2 kilos per day. This is before the epidemic. This is after the epidemic. 7th of August was the absolute champion. We have over 100 dog poos on the beach, as you can see here, which is about 8 kilos of dog feces. On the average is about 3 kilos a day, so that is about 1.3 tons of dog feces on this strand per year. Why is this important? Well, one thing is that dog feces has a much higher concentration of fecal indicators than human feces and sewage. So that means that if you put one dog poo on the size of a tennis court, 0.5 meter deep, and I've used this before, as an example, this water quality would fail, assuming homogeneous distribution. The other thing, of course, is that you can have disease-causing microorganisms in it. And finally, if you look at the location of these things, they're very close to the compliance point, and of course you don't have homogeneous distribution. Locally it can be very high. If a local authority would come in, take a sample on top of a dog poo, you get a failing water quality. So, and we know this is not hypothetical. We are testing water for the local authorities in the bay whenever the water quality is not excellent, to look at source tracking. And many of these that have lower quality of water do have high levels of dog contamination in them. So that's quite serious. Now, we've done a campaign, so we can say, what can you do about this? So in 23, we had a Leave Only Paw Prints campaign. You can see the reference here. They got a lot of attention nationwide, and an abridged version of this animation has been put on the entry points to certain beaches to educate the people. This is Niamh Martin, who was leading this, and here you see the team. You can see nicely lined up to analyze the dog poo. So very low tech, but very useful. So then you come to, okay, but this is from data from 2019, so you can say, okay, well, what about the contribution of each source in this part of the bay? So now we have evidence to see, okay, what is contributing. This stream is the main polluter, as you can see, under different conditions, maximum and lower conditions. Dogs can account for about 1/3 of the contamination here. Bear in mind, the contamination can be extremely local. And then, of course, wild birds, who have often been accused of the main culprits here. Birds have almost no impact at all. This was published last year. So if we look at safeguarding water quality, we need to know about the location of contamination sources. Where does pollution come in? We need to have the biological source of it. Is it a bird, a dog? I only talked about urban, but in agriculture areas, you have sheep and cattle. It's also wildlife. Then you need to quantify how much is coming from each source, and you need to look at the connectivity. If you release something over there, does it have an impact over there? Now, only then can you do evidence-based strategic interventions. So, for example, and this has been done, the Elm Park stream is much cleaner now. Irish Water has looked at the CSO and improved it. We have lots of pipes coming into this stream. The Leary County Council has looked at misconnections and found some of them, so that is a thing. You need behavioral interventions. Public doesn't know how important dog pollution actually is. They think it's a big beach, little poo, it will wash away. Well, it won't. And then, of course, if necessary, you need enforcement. So if things can't change and there is a way of enforcing it, then you need to do. So that is based on water quality, and basically fecal contamination is bad. Now, Aisling also asked me to talk about something else, and that is to say that sewage is good. So this is from Victor Hugo in Les Miserables. The sewer is the conscience of the city, and we can learn a lot from sewage. Now, this is related to the recast of the Urban Wastewater Treatment Directive, and you're probably familiar with this, at least some of you. It does a lot of things. What we are interested in is Article 17, that is to monitor wastewater for health parameters, SARS-CoV-2, other viruses, antimicrobial resistant, drugs, and so on. Now, why is this important? It is because we have, of course, info from clinical data, so notifiable diseases, hospitalizations with drugs, overdoses, unfortunately also people who die, so postmortems. This is surveillance as it usually takes place. This is wastewater-based epidemiology. And basically what you can look at is a range of readouts, as you can see here. We can do this in asymptomatic individuals, so we capture everybody. There's no bias. If you're sick or not sick, we can find you. And of course, with drugs, it's very important, because if I would do a survey here, ask everybody of you, have you used drugs last week? It's probably 100% no, because people don't want to admit this. However, wastewater doesn't lie, so therefore we can do this. It is very cost effective. Single samples will give you the info on a community. The community can be very small, and we've done this on a campus or even a meat factory. It can be a citywide, it can be countrywide, or it can even be global, so scalable. Now, these two approaches are complementary. It's not one replacing the other. They match. Now, this is what we have in Ireland, the National Wastewater Surveillance Program, which was established in 2021. It was preceded in 2020 in our lab immediately after the start of the pandemic, so in May, where we started with 3 wastewater treatment plants. It has since been expanded initially to 68 plants covering the entire country with an 85% coverage of the Irish population. We've scaled it down because we have more targets and fewer plants. Now we cover 70% of the population in Ireland, and data are reported back to the HPSC on a weekly basis. And this is governed by a steering group of HPSC, HSE, the National Virus Reference Laboratory, Irish Water, and of course UCD. So these are the people, the groups that are running this program. So the National Wastewater Surveillance Program that is currently in place, this is the website for it on the HPSC site. This is the link to it. We have a number of readouts, so SARS-CoV-2, a number of respiratory diseases in addition to SARS-CoV-2, influenza and RSV. Polio virus, especially the vaccine, the vaccine strain SARS-123, and the vaccine-derived polio virus, which fortunately we haven't detected in Ireland yet. And then there are a number of projects that are related to this, but funded through different sources, like antimicrobial resistance, metagenome analysis, illicit drugs, and avian influenza. So that you can get from sewage. This is how it works. So you take your samples from all over the country. We process this, extract in this case the RNA, in some cases also DNA, RNA, run qPCR, digital PCR, or in case of drugs, LC-MS. And then we analyze it using different sequencing platforms or indeed with mass spectrometry. And these are some details there. Now, this is an example, so I don't have time to give you a comprehensive overview. What you see here is the data from Ireland, total Ireland for SARS-CoV-2. Take initially 68 plants and then later 30 plants. On the y-axis, you see the distribution of the viral concentrations per week. So these are epiweeks coming from 2021 until this is September 2025, so the full duration. And if you follow the spine of this snake-like structure, you see the levels of SARS-CoV-2 in Ireland and the max and lows. And you can see that SARS-CoV-2 initially started off as a peaking at Christmas. And for the last 2 years, it has peaked during the summer times and actually reaching very high levels, including this summer. So that is what SARS-CoV-2 looks like. I don't have time to show you the other readouts. Another one that might interest you is you can also do this for drugs. We're part of the SCORE network, which looks at 37 wastewater treatment plants in 37 countries. And 120 cities are compared during a week, taking samples every day, and then you can have an impression of what's going on. And this is then what happens. I can't show you all. This is an interesting one. This is MDMA, or ecstasy. And if you ask yourself, is ecstasy a party drug? Well, it is, because during the week you don't see much use, but then during the weekend, ecstasy use spikes up. Whereas other drugs like marijuana and amphetamine remain flat, so these are habitual users. It also shows you the reactivity of the system, so you can quite accurately measure. And Dublin, you might be interested, we rank 19th of all the cities that were tested in ecstasy use. So this is just the people that are involved, so from our UCD, the HPSC, NVRL, Irish Water, and then here are the good people that are funding us, and this is the team. I would just like to point out before we leave David Kay, who was part of the Climatize project. Thank you.